To have him back. I'm very excited to introduce our main speaker today, Dr. Ken Hayes. Ken is a conservation biologist and molecular systematist specializing in Pacific Island biodiversity, invasive invertebrates, and the use of molecular tools to support biosecurity and conservation management in Hawaii. And Ken's co-author on today's presentation is longtime collaborator, Dr. Norine Yeung, a conservation biologist specializing in Hawaiian land snail ecology, taxonomy, and invasion biology, with research focused on the impacts of invasive soil organisms on island ecosystems. And as I mentioned in the talk that Ken gave last week on invasive worms, Ken and Noreen are the go-to experts for snails, slugs, invasive worms. Anytime we get a new or unusual worm or slug or snail report that comes into our 643 Pest online system, they're our go-to indispensable experts on this topic, and they're invaluable assets to Hawaii's biosecurity. So want to thank them again for presenting today. The title of the talk is Malama Kahuli, Leveraging Systematics for Biosecurity Stewardship in the Age of Global Molluscan Invaders. And with that, I will say thank you, and please feel free to take it away, Ken.
All right. Thanks, Chuck. I appreciate it. And thanks again, Beth, also for the invitation and the opportunity to share some of the work we've been doing to help sort of Malama Aina. All right, so just to kick it off, right, place it into our biocultural sort of framework as well, which we always like to do here in Hawaii, to understand that we have this immense biocultural treasure, not just the plants and animals, but the culture that sort of defines it and relies on it, right? And the problem this sets up invasives, as you know, a crisis of ecological, but also cultural extinction. So whenever we lose one of these beautiful voices of the forest, these snails that were purported to sing in the forest and be good omens, we're also losing that cultural connection. We're losing that voice of the forest and our ability to better understand it, right? And as I mentioned, you know, the mele and protocols, as well as other traditions, speak of the Hawaiian land snails as singing in the forest, right? They're voices of the forest, and they're spiritual icons, celebrated for their ecological balance and omen of good things to come, right? Ecologically, these microvores are unusual in that they perform vital tasks like micrograzing of bacteria, fungi off the leaf surfaces, and it helps accelerate nutrient cycling into these forests, which are so critical that I talked about last week, right? And what's happening is we're getting this rapid silencing of our woodlands, right? Our upper forests, right? We historically had more than 750 species of endemic snails. And today, you know, the habitat loss due to clearing and habitat destruction, but the loss of their plant host as well, and then invasive species have driven at least 50% of these to extinction and more for some of the families, which is irreversible, right? And these are the groups. These are the 13 families that we recognize taxonomically here in Hawaii. And you can see the numbers of species that have been described and are recognized as nomenclaturally valid. So in the Acatenellidae, we had 215 originally. The Amastridae, 325. And I should mention the Amastridae is family that's endemic wholly to Hawaii, the entire family. And until recently, it was the only entirely endemic family in the islands. But the other 11 there you can see. And what's shocking is when we look at this and we talk about extinction is these are sort of our estimated rates from the last, say, 15 or 20 years that we've been doing this of the percent extinct in each of those families. Now, these numbers are lower in some cases because we've included some of the undescribed species that were in there. So those numbers go up. So they're not a direct reflection of the described already, but again, pretty close. And again, this sort of omen of biocultural extinction is not great. And it's driven heavily by invasive species, which is what we're going to talk about here today. So what are the invaders, right? These sort of pests that we consider them, or unwelcome guests that damage our environment. And then you see the headlines again across the globe, but particularly in the US and here in Hawaii, even. Snails really capture the headlines when they are detected, often after the fact, once they're damaging. So you can here see an article from 2021 of how much money Florida spent trying to eradicate African snails. So, we're very familiar with them and the damage they cause. And Florida spent 24 million. I think by the time it was all said and done, it was more like between the Florida and the federal government, they'd spent about $120 million trying to eradicate it and declared it a success, only to have a month or two later it pop up in another place. So that tells us it's really difficult to get rid of these things once they get well established. So we really need to be on guard to try to detect these as early as possible. So they bring diseases with them. All right, here's a headline from a news article just last year talking about 150 diseases spread with mystery snails in Missouri lakes. And again, the species they're dealing with there is a viviparad, a species we also have here on Kauai that's been established. And then, of course, zombie snails, right? Those headlines make great, but what they're talking about is the parasitic flukes that occur, or flatworms that occur in these snails that infect the snails and make them behave a certain way, but they can also be harmful to humans. And there's a number of reasons they get introduced. So there are purposeful introductions, right? So here's a couple of the headlines pulled together over the last few years. This woman that smuggled through French customs snails in her brassiere, right? Live snails, get it? Pretty brazen. Must really want snails pretty bad. But South Florida, again, giant snail smuggled for its slime. This is how the African snail was originally introduced to Florida. Most recently, because a religious group was using their slime in religious practices. And then this one here, this guy smuggling 100 snakes in his pants, right? It's pretty bizarre in purposeful introductions. But I'd say by and far, most of them are probably accidental introductions coming with plant trade or other trade. They come as agricultural pests, and that's probably the thing we think of mostly when we're thinking of invasive species, but they really do alter host plant and animal pathogens. So they bring those with them as well. And that little icon there in the middle is to remind you that when we introduce a species, not just snails, but anything, they don't just come as themselves. They come with a whole host of their pathogens and parasites and things like that come with them. And so these can threaten our native ecosystems as well. And I want to think about this, there's a lot of talk about the One Health framework for human or public health, but really getting to the side of animal and plant health, the non-animal or non-human animals, is really important to understand this because we rely on these so heavily. And I really want to promote this idea that understanding our ecosystems and the animals that occupy it really will help our own public health as well. And I showed a bit of this last week, the more than 2 centuries of introduction trends, right? Showing where they're coming in and why. And again, it's mostly container freight coming in with various commodities. And you can look at the literature and see nurseries, horticultural products top the list almost every time, every survey, everywhere they've ever been done. The military is pretty close behind. A lot of the things that they move around carry lots of invasive species. And then, of course, just sort of odd ones like things like tile and other commodities that come from different parts of the world. And again, it's correlated with that GDP. The more stuff you import, spend, the more invasive species you're going to see. And again, just a reminder, we often think about invasive species or the impacts on biodiversity in these ways, right? So with overexploitation, invasive species, climate change, and habitat destruction all working synergistically to cause alter the ecosystems and cause biodiversity loss. But a bigger issue is our knowledge shortfalls, the things we just don't know. And without the knowledge, filling these knowledge gaps or knowledge shortfalls, we really can't address these other things. We can't really do anything about invasive species or habitat destruction, overexploitation, without an understanding of biodiversity writ large and the components of it that are really important. All right, so to address this for a number of years now, more than 20, our team has been doing surveys for a wide variety of habitat types. We've over 2,865 to date, with about 10% of those specifically targeting agricultural and horticultural sites. And this is filling one of those shortfalls, what we call the Wallacean shortfall, or a shortfall in understanding the distributions of species and where they live. And once we gather those data, we then use an integrative taxonomic approach that combines shell morphology or conchology with modern genetic approaches and genomic analysis, as well as traditional anatomical analysis and biogeography. All of those combined, this sort of multi-field approach to really kind of understand what's going on, what we have here, and how they're spreading and what impacts they might have. And if you look at the last 2 centuries in Hawaii, that same sort of hockey stick pattern shows up that Hume also saw globally, probably driven by the same thing, trade and such. So you can see, here after the 1950s, things start to pick up considerably. But towards the end here, you see the introduced species and established species, particularly the last 3 decades, really tick up fast. And that isn't really a signal that things are coming in more frequently, even though they might be. It's actually reflects our survey period, right? Our sort of effort to do other than just opportunistic surveys, but doing targeted surveys to detect these things. And that's really where those data come from. And what you can see is it's really jumped up over the last 2 decades, with about half of everything we've detected as introductions becoming established. And I'll talk a little more about that in a minute. But this is the complete list as of 2026, as of this month. We have about 36 families and 89 non-native species that have been introduced. And you can see the ones in blue indicate those that are freshwater. So these are non-marine species, not just the terrestrial ones. And they have a wide variety of impacts. And I'm going to use this baseline as to highlight a couple examples of how we approach this and how I think that biosecurity could benefit from implementing more of this type of approach across the board. Right? And again, when you start looking at the pathways in the literature, not just here in Hawaii, but everywhere, again, more than half of all studies have shown that the horticulture and plant trade is by far the largest contributor to these non-native mollusks coming in. Aquaria and pet pathways, so this is the pet trade. This is mostly freshwater stuff, but some terrestrials as well. And then, of course, agricultural freight, which they distinguish from plant trade by agriculture. So if you combine these two together, we're looking at 60% of all the mollusk species being introduced are coming from horticulture and agriculture. And then, of course, a major contender there, and it's actually a low number, relatively speaking, because of the level of inspection that happens there. It doesn't happen quite at the same level because of the military prevention of that, right? They sort of manage their own. So we don't always know. And it's when you have been able to look, it comes out at about 10%. So a few case studies using this integrative taxonomy here to deal with species in Hawaii. Start with one that's on the forefront. These are 2 species that were detected 3 or 4 years ago in Hawaii, maybe even a little bit further back. At the top right, you have Zacrisia professoria and another helicid down here, Otella lactea. Both of these are really nasty invaders where they've been introduced to consuming massive amounts of plants and reaching really high densities. This one up here, we've suggested that it will be as bad as the giant African snail, if not worse, once it gets established and spreads. This one can be very, very bad. So that first one, that Cuban brown snail or Zacrisia professoria. So a while back, we were contacted in much the same way that Chuck was mentioning earlier by the 643 Pest, or I can't remember if it was Janice when she was at H2A contacting us. But anyway, we were contacted because a resident had found these in their backyard. So this is a picture of their backyard here, that they were growing plants in these half tubs, and they had found this snail. So this is actually a picture of them there. So we came out, did surveys, and did a provisional identification and said, oh, yeah, that looks like the Cuban brown snail, caminid. What's interesting is if the house is in this neighborhood here, right across the street, this is a native plant nursery, right? That proximity terrifies us, right? Because this is the pathway. And this is called the Cuban brown snail because it lives in Cuba, which has a lot of the similar habitat that we have here. So it's a real potential for a fast spreader. And this is what we told the HDOA and others, that we need to find out where they got this from. I took it back, a couple of the specimens we took back, and we notified the USDA as well. And just note that this is a quarantine status in it's non-actionable in mainland US, but in Hawaii and Puerto Rico it is, because it wasn't introduced. Now it is here, and this was the only location. So HDOA, with the help of the resident, tracked down the nursery where this was, he supposedly had bought materials from. Went back there, sure enough, found multiple size classes at that nursery, and at the time chose not to shut it down for a variety of reasons. And we warned that it was going to be a matter of time before you see this spread rapidly. And sure enough, within a year or so, we saw it in multiple places on Oahu, and most recently it's been intercepted on Lanai, and it's only time before it ends up on the other islands. But the identification can be confused with these. So this is Acrisia provostauria down here, and these are two other species in the same genus. So these are some generics that it could be confused with, also equally damaging. But these are pictures of what Acrisia can do to crops. It's quite damaging. There's a list of over 40 or 50 crops that it does impact, and it will definitely have impacts on native vegetation as well. The other one that we reported about the same time is this one. This one was first reported to us through the HDOA again, but more recently, a volunteer with us had found this in her backyard shortly after moving from Florida, and brought it in and identified. What's interesting was the first report was actually from the HDOA. You can see some of the pictures they provided here of a car that was imported from Europe, but it laid over in, I believe, San Diego in a base yard there waiting to be exported. And this is probably where it picked it up. It picked up this Otala, which climbed up in here. But you can see from this article here, this Australia, right? The Mercedes-Benz also had it, right? Same kind of pathway. So this is not a uncommon pathway, but still sort of unusual for us to see. Luckily, this one was in a showroom inside of a building when it started coming out, which is interesting. So they were able to contain it, and it doesn't appear to have spread, fortunately. But again, same thing. We took it back, and using shell morphology, some genetics, as well as the anatomy, we're able to place it into its proper species group. And again, same thing, quarantine status with USDA. It's actionable in Hawaii and Puerto Rico. So mandated to do something to get rid of that quickly if you can. And this is kind of what it looks like when you can see a whole ton of them, right? They quite variable, but can be easily distinguished by that black lip from some of the more closely related individuals, the inside black callus lip. But you can see here they can map huge numbers, right? So this is from Los Angeles County, and they can get it to huge numbers. And this is where they cause a lot of damage and problems, particularly in places like Australia and other where they've been introduced and become established. And like I said, it can be confused with this one, the chocolate band snail, Embonia vermiculata. And again, notice the absence of that black lip, right? But again, same kind of behavior. They have both of them in LA, and just massive aggregators. And I don't know if you've ever seen giant African snails tend to do this too, and used to do it a lot more here in Hawaii. They climb up on things to get out of the heat, and then go down at night to feed, and can even feed on plant crops. So our papayas and other fruit trees, mangoes, things like that are definitely at risk from these invaders. All right, switching gears from species that are mostly restricted to, different parts of the world, to a family that has sort of really wide distributions, the Succineidae, often affectionately called snail, snot with a hat here in Hawaii. And we have about 36 to 38 native species here in Hawaii. But these are the two that we primarily thought we had here in Hawaii for the longest time. So there was Succinia tenella and Succinia costaricana were the two that were most often identified. But as you probably learned from my other talks and things that we talk about, is that it's never quite as simple as that. And succineids are notorious for being difficult to identify at the species level because their shells are almost impossible to tell apart at times, and they lack a lot of reproductive material to characters to help you delineate them. But again, we used an integrative approach to combine this conchology with some biogeography, but also looking at museum specimens. And that's a really important part of this. So not only do museum specimens provide foundation for your taxonomic decision making, but are also critical in understanding the timeline of invasions. Lots of times, if surveys are being done regularly, these would be picked up and maybe misidentified, but then can be sorted out later. And I'll give you an example of that in a bit. But we published this paper in 2025, and we entitled it Sorting Out the Stupefying Systematics of Succineae Introduced to Hawaii. And what it turns out is one of the two names we had was correct. So Succinea costaricana was correctly identified, but turns out that the thing we were calling Succinea tenella was two species, one being Succinea unicolor, and that's probably the most widespread one here in Hawaii. It's occurring on all of the islands. And then Luteola, which is a more recent introduction, but also misidentified. So we completely missed this introduction and spread in establishment because it was misidentified. So a real good example of how cryptic species, cryptic diversity can sort of let things slip through the cracks. And so we really need to do a better job of our revisionary taxonomy to make sure we're dealing with the same things when they come in. And we really got to get better at monitoring the pathways, not after infestation, not once it becomes a problem, but ahead of time. Because as you can see from this picture here on the left-hand side, they can be very difficult to see, right? So it's obvious there's one there, but here's some eggs down in here, right? And eggs there, another species of snail there, another one there. There's a couple more in here as well. So they can be very difficult to see, especially when they're in low numbers, but it doesn't take long for something like this to spread. And because it's very difficult to identify them just by shell morphology alone, it often requires some broad taxonomic training or really integrative approaches to clarify that and help understand those pathways. So switching gears from terrestrial to freshwater, we see, again, a group that was thought to be well known. So the Thiards have been in Hawaii for, prior to this work, thought to be, prior to Polynesians, there was thought to be species that were endemic. But we put this paper together after a lot of, an enormous amount of work, a lot more work than we thought it would be to clarify this. But just to remind you from that graph earlier, we had 6 families of freshwater species. And we thought at that time, maybe keeping the Thiards as there were at least 3 species of Thiards in Hawaii and a handful of others. So about 19 freshwater species in total. But again, the situation is always less simple than what we think it is. So in 2024, this is kind of what the picture looked like with the different species of Thiards in Hawaii. And so I should mention Thiards are a notoriously invasive group. And again, another group that is really difficult to identify based on shell morphology alone. And that's partly exacerbated by the fact that these things many of them are parthenogenic, meaning that a single individual can give virgin birth. They don't need to outcross, they don't need sperm, they can just create babies as they go. And almost all of the Thiards, particularly those in the Melanoides group, usually have several babies in the chamber, if you will, at a time, dozens of them, and are ready to pop those out as soon as they have the opportunity. So they're the right mixture of traits to make them highly invasive. They can also tolerate incredibly disgusting water. And once you get them, it's almost impossible to get rid of them. But these are the species that we were thought to have in Hawaii with their locations of where they were first described from. So you can see Melanoides tuberculata. This is a red-lipped melania. Originally from India, possibly that's where its type locality. It's a valid taxonomic status. Its first record in Hawaii was 1994. That doesn't mean that's when it first got here. It's just the first published record. Scabra, we reported in 2025. So technically, I guess this is as of 2025. Tribia granifera, it's from West Timor, and again, valid. It was first reported in 1857, but as Mauiensis by Combe. We had Mauiensis from Maui. Again, you can just look down this list. And so we're going back well over 100 years of records here in the Hawaiian Islands and a bunch of different species. But these are the ones we considered valid in terms of what's really here, right? Looking at 1, 2, 3, 4, 5, 6. The rest of these had basically been considered as synonyms of one of these other species that are valid. So either Melanoides tuberculata usually or Toribia granifera. Most of the Thiara had been put into Toribia and other things have been into Melanoides or Melania. And why is it such a big problem? As I mentioned, they can reach really high numbers. They can survive almost anywhere, but they are notorious as carriers of various parasites. And here's one that we recently detected in a group, the Philophthalmus species. This is a flatworm or trematode parasite that affects waterfowl primarily, but it's an optic parasite. So it gets inside, as it develops, it gets inside, and the adult gets inside the eye of the host. So sometimes it can get into humans as well. So by eating infected birds or even infected snails, right? And so the snails is a critical component of that life cycle. So it affects our native waterfowl as well as human health. And again, like I said, we tackled this by surveying lots and lots of sites. That's about 1,700 at that time, looking for these freshwater species. We had examined 132 lots from the museum, going back well over 130 years or so, and about 720 specimens from 97 different sites. And we used shells of the recent historical and type material to delineate these, and then sequences from another 344 individuals, spanning not only Hawaii, but lots of other places in the world where it had been introduced. And then we used a phylogenetic analysis as well as some Bayesian species delimitation techniques to match these up, because again, the shell morphology can be quite problematic. This is our phylogeny here. This is the concatenated one with 4 different genes, spanning all the taxonomic groups. And the first thing you pull away from this is you see in blue are all of the Hawaii groups, right? So it's not, as we thought, just a simple couple species, 3 or 4 species introduced. Turns out there's multiple species and many of them that we can't even really identify properly. When you look at it closely, right, you see we've got 4 of these up here, that Menpleota scabra, which also is non-monophyletic because you've got some individuals from that same group that fall outside. And then you've got a number of Melanoides species here that are in Hawaii, including Melanoides tuberculata, but 2 things that we left in open nomenclature because we couldn't really put an ID on them yet. Again, this family has more than 300 species in it, so it's a group that really needs to be revised globally to make sense of this. And those are nested in there. But you look at that second clade, which is further down here. This is where our Thiara and Toribia, right? And Thiara hawaiiensis, I should mention, is one of these that was thought to be a native Hawaiian species, but turns out it's not. It's a very widespread thing, and this analysis indicates it's not native at all. And so we don't have any native Thiarids here in Hawaii, even though they're highly agile species in terms they tend to move around. And it turns out, going all the way back to fish pond records of these cores from the 1800s, and when Polynesians first got here, they came with Polynesians. So some of these, like are technically canoe snails that came in very, very early, but they're not native because there are no pre-Polynesian records of them. And like I said, once you're able to use some delimitation analysis and really track down type material in museums around the world, you're able to make a little more sense of this by looking at the shells. And the shells start to look quite distinctive, even if they are quite variable. But it does take quite a bit of work to really get at that information. As you can see, there's quite a range of variation in shell morphologies in all of these species. Okay. So again, we had these previous identifications here in Hawaii. So these 1, 2, 3, 4, 5, 6 species. And what we end up with is 1, 2, 3, 4, 5, 6, and 7 species total, technically 8, one that's only a shell that doesn't show up. But again, and we were able to clarify some of these other names that were in Hawaii. So this one's a nomen dubium, meaning that it's a questionable name that couldn't be reliably assigned to anything, so it's not a valid name. And so these are the species that we currently have in here in Hawaii. Some of them more widespread than others, some restricted to just the aquarium trade or the aquatic plant trade, and a few that are in our high elevation habitats all the way up into the streams with some of our native species. So quite a number of different impacts possible there. And really the take home from all of this, right, with just a few of these, but the same message applies, is that, we have lots of cryptic species, 3 in Hawaii, and then 7 widespread species of thearids in Hawaii. And Theara indefinita, that's one of the ones that, was historically recorded here, but we only found shells of it most recently in the museum. And so it hasn't been seen in our recent surveys. But again, no native thearids in Hawaii, and some possibly canoe snails. So we've got a mixture of things, that families that are native here with non-natives invading, which is problematic as it creates the possibility of crossover or spillover of parasites and pathogens, but also competition for habitats. These aquatic ones are really problematic. And again, the biggest take home from all of this, as I keep harping on, is that taxonomy is not optional, right? Misidentifications are muddling our understanding and distort the ability to understand invasion biology, parasitology, species interactions, ecosystem impacts, and ultimately resource management, right? And really need to make sure funding, as it comes down the pipelines, target 3 really broad areas. Proactive surveys. So let's not wait until the thing's already infested and established and causing damage. Let's get it before it gets damaged, because we can understand that using an integrated approach of how much effort we need to put into eradicating it. Not all invasive species cause the same level of damage, but you can't know that if you can't identify it. And of course, taxonomic training, because, Chuck had some really nice words for Norine and I, saying that we're indispensable, but as much as we like being indispensable, we also don't want to be the only people, so that when we retire or stop doing this, there won't be anybody left to do it. We're really desperately trying to train another generation, but one of the ways we can get this is making sure everyone's broadly trained. So we offer taxonomic workshops and different types of tools to bring to bear on this. And having funding for those kinds of things is really important. Mostly we do it for free, but funding really helps propagate it much more effectively. And then, of course, we've got to fund revisionary science. So this is just research in general. We can't always be reactionary. We have to fund the revisionary science needed to do it. And this is an area that hasn't been funded for many decades. People think taxonomy is boring, and you're hard pressed to find real taxonomy focused courses at the university anymore. And that's because nobody wants to be a taxonomist, not even Norine and I. Neither of us ever said, oh, I'm going to go to graduate school and become a taxonomist. It's just that we ended up working on groups that we quickly realized that, well, if you want to answer interesting questions, you got to actually be able to identify the organisms accurately. Otherwise, you're misinterpreting data. You're mixing 2 species, or you have one species you lump out as 2 or 3. It really creates problems, right? And so you have to have the revisionary basis to make these kinds of interpretations. And so that's, unfortunately or fortunately, however you want to look at it, it's where we spend a lot of our time, is doing revisionary work to just identify these things. And then try to make that data available. And again, which all of it costs money, right? And we need to use this kind of idea to modernize biosecurity frameworks, right? So these functional foci down here, so primary diagnostic tools. So if you look at the traditional pathways, traditional ways we did this was primarily with mollusks, you looked at shell morphology, right? And you compared them, and it was a mostly visual a tool, right? Whereas nowadays we tend to rely heavily on integrative taxonomy, and you can get molecular data pretty quick. That doesn't mean just rely on barcodes, because barcodes can be misleading as well without the framework to interpret them. Interceptions, right? These tended to be reactive, and they still tend to be reactive to some change. We're responding to damage or responding to finding them, as opposed to doing what we call sentinel surveys, just constant annual surveys in different hotspots or areas where we know these things are coming in, to make sure this doesn't happen. And I know this is partly a function of funding for labor, right? Having the staff to do it. But I think partnering with researchers and others to do this is a real effective way to get it done, to spread that funding around. Species delineation, right? Again, the traditional approach of looking at shells or just gross morphology is really prone to misidentifying cryptic complexes. And our integrative approach really helps resolve these lineages and better understand where we can most effectively apply mitigation efforts. And then the funding architecture, right? So funding architecture historically is post-invasion mitigation. Let's find a biocontrol, or let's find some way to eradicate it. This is very expensive, right? This is a waste of money. If we would put more money up front, we could save money on the back end. And this is a much more cost-effective way of doing things. Pre-invasion training and implication is really critical, right? So the biggest point I like to make is we cannot protect what we cannot identify. So this goes for native species, non-native species, and funding of alpha taxonomy and continuous field surveys are not just merely academic pursuits, which is the way they've been treated for, I don't know, at least half a century or more since World War II. And these are actually the primary mechanisms to operationalize Malama. We can't take care of the land, right, if we're not connected to it. And Hawaiians understood this. They were deeply connected to their land out of cultural respect, but also out of necessity. Hawaii's not a hospitable place to live if you don't understand how to manage the resources that you're living off of. So they did it rather well, and there's a lot of lessons we can learn from that. But if we want to operationalize Malama, we need to do a better job of identifying things and surveying for them. And I mentioned this last week, taking sort of this cancer biology approach, right? And applying it to biosecurity, looking at pre-border prevention, border detection, post-border response, biocontrol and containment and rehabilitation. And these are the areas up here where we're going to be most successful. Down here, it gets more expensive. It's better if we can target it up here. And again, looking at these different ways of handling it: taxonomic training, pathway intervention, outreach education, collaborative monitoring, and research, all the same. And again, taxonomy is important at every one of these levels to help us address all of these major knowledge shortfalls that we have. And as I mentioned, we produce guides, right? Norine and I are all about, and our team are all about sort of broadly disseminating this information wherever we can, in any way we can. And so these are the guides to aquatic freshwater snails, introduced snails, slug guide. We separate slugs and snails out because people like to have them separated out. But just be clear, slugs are snails. They're not a different taxonomic group. Slugginess has evolved more than once from multiple groups. It's just a habitat sort of modification, adaptation to deal with desiccation differently. And so, again, we separate them out to help you identify them. And these can be downloaded on one of the sites that we have made available for these. So if you want those, click on that QR code or scan that QR code, and it'll take you there to fill out a quick information form just so we can keep track of who's using it. These are primarily funded through previous HISC proposals, but also USDA as well as the National Science Foundation have helped us keep these developed. And as you can imagine, with that graph of species continuing to increase, we continue to get new introductions. These are only good for short periods of time. They have to be continually updated, right? Because it doesn't help you detect other things. What it's useful for is in looking at them, you'll be able to tell, like, oh, this doesn't look like anything on here, or one of the ones we've talked about. So it can be helpful in that way, but they are constantly in need of update. So with that, there's always a ton of people to acknowledge and thank, particularly those doing all the hard work, boots on the ground here in Hawaii, as well as our colleagues at the USDA and elsewhere. And then all the various funding agencies and groups that we participate with. And last but definitely not least are our crew, the colleagues we've had the fortune to work with over the years that helped enrich the surveys, but help enrich our understanding of it, and that we get to spend good quality time with in the field and in the lab.